Thanks, Tammy and Sarah, for having me, and thanks to everyone for coming this morning. So Sarah and I are going to discuss our work prior to our collaborations, just to give you a flavor about what we do outside of our work together, and then Sarah will go over for most of the talk our collaborations together. So what I'd like to do is talk to you about, yes, that's us when we were little. So my background is in cognitive neuroscience, and the focus of my research is to determine mechanisms that underlie symptoms of addictive behaviors. So my real interest then is to determine biomarkers so that we can then better predict who might be at risk for addiction, but also be able to target the treatment that might work best for them. And that's when our interests, Sarah and mine, match perfectly well. The type of neuroimaging tool that I use most typically is MRI, and that's the focus of my talk today. So with MRI, we can collect data on the anatomy of the brain. Also, we can determine how white matter structure looks like using diffusion tensor imaging, and then we can also look at how the brain functions using functional MRI. So I'll just briefly touch on some of the work that I've done using these techniques, starting with structural MRI. And most of what I'll show you is on marijuana users, a population of daily users who've been using for at least 7 years. Their mean age is about mid-20s. In this study, what we wanted to do was to determine whether there were structural differences between chronic users compared to non-using controls. And what we found was that an area called the orbital frontal cortex was smaller in chronic users relative to controls. This area is important for adaptive learning and also for motivation. And so that really piqued our interest, and we wanted to investigate that further. And when we did that, we found that unlike the reduced volumes, what we found was that there was increased connectivity. What I mean by that is when we looked at the white matter that connects the two areas of the orbital frontal cortex called the forceps minor, what we found was that this white matter tract, the integrity of that tract was actually greater or higher for the chronic users relative to controls, which really surprised us because for other substance abusers, this is not typically the case. So you can see here, although not very well, when we graphed the fractional anisotropy, which is an indicator of how water molecules travel down the white matter paths, you can see, again, not very well in green, that the marijuana users had higher FA, which again indicates better white matter connectivity relative to the controls, which are here in blue. And we also compared them to nicotine users, which are in red. As you can see in that tract, marijuana users had greater FA relative to those other two groups. When we plotted the FA against years of use, so duration of their onset of, or duration of their regular marijuana use, you can see that increase in FA during initiation of use all the way until about the 4th or 5th year of continued use, and then that pattern starts to decline. So what we're seeing here is a quadratic relationship between that increase in white matter connectivity and years of use, suggesting that perhaps during prolonged use, after about 5th or 6th year of use, that FA connectivity does start to decline. We need to really determine if that might go back to the level of the non-using controls, but it suggests some type of neuroplasticity perhaps that occurs immediately after the onset of regular use. This reminds me about what Laura Ray said yesterday, that we really need to determine the patterns that go along with the different phases of addiction. This indicates that we could really tease apart the differences that occur in mechanisms that occur during the transition towards dependence. So the orbitofrontal cortex is one of the key regions in the dopaminergic reward pathway. And so functionally, we were interested in determining how this reward pathway of marijuana users might be altered compared to controls. And so to really examine that, we wanted to look at reward processing using a cue-elicited craving task. So for this group of marijuana users, we wanted to see how they might respond to a marijuana cue, such as a used marijuana pipe. What we did while they were in the scanner is we handed them this marijuana pipe, so they were holding it. Again, it was used, legally acquired. So it also smelled like marijuana. And they also viewed, the mirror was positioned just over their eye so that they could see their own hand holding the pipe. So it was as ecologically valid as we could make it. And we wanted to see what the response to that experience was relative to when they're holding something completely benign, completely neutral. And so we used a similarly colored pencil. Following that cue exposure, we also asked them to rate their subjective craving or their urge to use marijuana right that moment, and then followed by a washout period. So what we found when they were holding that pipe relative to the pencil was that there was greater activation almost in most of the brain areas. I have highlighted here key areas that I was very excited about. That includes the VTA, ventral tegmental area, the orbitofrontal cortex. See some insula activation there as well. In addition to greater response to the marijuana cue, we also found that this response was positively correlated with the amount of problems they have with marijuana use using the marijuana problem scale. And it was also negatively correlated with age of onset. So the younger that they started their regular use, the greater the activation in this area. And with regards to the problems with marijuana, the greater the response in this area, the greater score on the marijuana problem scale. So the question then becomes, how do they differ from normal controls? So non-using individuals. Perhaps non-using individuals would also respond similarly to this highly novel object, and maybe that's all that we're capturing there. And also, how would it look like in terms of natural rewards, or compared to natural rewards besides a pencil? Because you would expect that, reward pathway would not activate or respond to a pencil, but it should to natural rewards. In theory, what that would answer for us is whether marijuana does hijack this reward system. And so we modified the task to include those elements. We also made it so that these reward cues were individualized, and so we asked participants to tell us what their most common paraphernalia is that they use. So we gave them some options of a pipe, a bong, a joint. And then for repetitive cues, we couldn't give them Cheetos or Fritos because we just thought that would definitely induce craving. plus it's not really natural, and so we thought maybe we should just give them, fruit. And in fact, we do ask them subjectively whether they like fruit, and we individualize it as well. We gave them the option of choosing an apple, banana, orange, and grapes. We kept it to fruits that people were familiar with and that were obviously accessible in our area. So what we found, first of all, we wanted to see how marijuana users differ from non-users in terms of their response to marijuana pipe relative to a neutral object, and we found that marijuana users, as expected, had greater response, and as you can see, in a pretty expansive area, relevant to non-users. Now, the question is, do non-users also respond the same way? Maybe, again, it's just the novelty of the object, and in fact, they do, but you can see that the difference is quite striking. So now, what about these natural rewards? Do marijuana users still respond to natural rewards? Again, the contrast is against a neutral object. And we found that they do. So they still do find these natural rewards appetizing, but visually, it looks a little less. In terms of extent of activation relative to the marijuana pipe. And for normal controls, it looks like almost similar amount of response. So suggesting that perhaps the response to natural rewards might still be within what you would consider normal in terms of marijuana users, but just a little bit more sense or a little bit greater for marijuana pipe. So we made that direct contrast between the response to marijuana pipe relative to fruit. Again, this is the group map for the marijuana users, and you can see that the difference is really within the front of the brain there in the orbitofrontal cortex and all along the anterior cingulate gyrus. Actually very small here, but you could see some striatal activation as well. So all key regions within the reward pathway. And then here's the group map for the normal controls. So normal controls really do not think marijuana pipe, or that they don't really respond to a marijuana pipe relative to the fruit. And then the direct contrast shows this difference. Again, those key areas highly different in marijuana users. So other areas that I have also been interested in is in terms of modulators of this reward response in users, stress being one of those, one of the areas that I've begun to look at. And so to do that, we use the exact same paradigm that I've just described to you, but then introduced a stressful event while in the scanner to these group of participants. So what we did to induce stress was gave them the Montreal Imaging Stress Task, which is an fMRI or an imaging version based on the Trier stress task. So what happens is that they have to perform a very difficult arithmetic test, difficult for the level that the participants can normally successfully carry out. They're given an amount of time wherein they have to perform the task, and then their performance is continuously monitored, so the bottom one being the participant's performance, and the top arrow indicating the performance of the average of our participants. So showing some social component, and also we tell them that they're constantly monitored in the exam or in the control room. And what we found when we also had a control task where we had all of these visual stimuli present, and they just have to press a button whether they saw a blue arrow or not, to try to control for any visual and motor elements. And what we found when we looked at the cue exposure period following a stressful event is that, again, I'm sorry, this is very small, but again, there's activation in the orbitofrontal cortex following a stressful event, but not following a non-stressful event. So suggesting that stress really, or acute stress, induces a higher craving in terms of neural response to cues. So with that, just to summarize this area of work, we have found that marijuana use does alter the reward system. In terms of structure, it reduces the volume of the orbitofrontal cortex. Again, we don't know whether it's the direct consequence of marijuana use because we didn't follow these individuals over time, or whether that was a predisposing factor, and so that's an area that I'd like to look at further. But functionally, that cue reactivity or cue response in these areas within the reward system was also altered, and that response is modulated by stress, and that it's also related to age of onset of regular use, and as well as marijuana-related problems. So with that, I'd like to hand it over to Sarah.
Thanks so much, Francesca. I realized I should have said before we started that part of the goal of Francesca and my talk today is to talk a little bit about our backgrounds as a neuroimager and a clinician, and then introduce our collaboration and how that worked. Just to offer one example of how you can collaborate with someone in a different field in order to get this kind of work off the ground. So I will start here by just giving you a little bit of background. Yifra Kaminer asked yesterday for us to think about adolescent marijuana use and how it's different. And so here today, I point out, historically, we all know here at RSA that alcohol use has been a huge problem for kids throughout the past few decades. So by the time kids graduate from high school, 75% are using alcohol, and about 40% are binge drinking. However, rates of marijuana are also increasing. So these are relatively old data from SAMHSA, but with recent changes in legalization, the rates of marijuana use are also increasing. And one reason for that might be changes in accessibility. So historically, rates of alcohol use have increased as kids find that it's easier to get alcohol as they're older, and this is some data from Oregon. So in 8th grade, it's less easy than in 11th grade, or the perception is that it's increasingly easy to get alcohol, and the same is now happening with marijuana. So right now, this is just something for us to think about with these changes. So historically, kids have been using 40% of kids have been using marijuana throughout the high school years, and that number is now on the rise. So I came to this field because I was really interested in how to help kids who are not receiving treatment. And what you'll see here is that kids don't self-refer. So I've been treating adolescent substance users for about getting close to 15 years now, and I still haven't had a kid come up to me and say, Dr. Sarah, I'm really struggling with my cannabis use and I'd really like to cut back. So I always think about adult studies, you guys get to bring in people because they want to change. We don't get that opportunity. So the kids we get don't self-refer because they don't often identify themselves as having a problem. And so that's something that we really have to wrestle with. So this is a really nice, another SAMHSA slide showing that there are 20.2 million kids who are using but not receiving the help they need to reduce their substance use. So my early days in my clinical development were under Bill Miller, and my goal was to take his empirically supported treatment and make it work for kids. So my first many years, and still to date, I spent writing clinical manuals, treatment manuals, trying to adapt MI for underserved kids that were in the juvenile justice system, as well as in other communities. And the real challenge in this work has been how to maintain the active ingredients of an intervention while making it fit for different communities. And the trick of that, which should be familiar to you MOBC folks, is that we don't totally know what the active ingredients of motivational interviewing are. So it's very hard for us to figure out how we can adapt it and make it work for different communities. So, and this is some very preliminary data. I looked to Angela Bryan, who is working on this data with me at the moment. So this is not the final version, but this is a kind of a first peek at this a huge R01 we just completed with 560 kids, where we gave them all 2 sessions of motivational interviewing. These are kids who are using alcohol and or marijuana and who are in the justice system. We gave them MI, and we tried to think, what's the standard of care that they get in the justice system? And that's alcohol education, where somebody comes in and tells them how bad everything is and hopes that that changes. And we thought, surely that won't do anything. It can't do anything. The data is so clear that won't do a thing. And so this is the 3-month outcomes, and so these are our proposed mediators. We thought motivation to change at 3 months and self-efficacy would surely, that treatment would change those 2 things. And what you'll see is it did not. You'll also see this very sad dark line of how there was no difference in treatment condition at 6 months for problem drinking. And again, this is our first pass at the data. Interestingly, motivation to change in that at 3 months did lead to reductions in problem drinking and in the direction expected. Similarly, the self-efficacy change, or variation in that led to reductions in problem drinking at 6 months. So we seem to have the mechanisms right, but we don't have how the treatment changes the mechanisms, or we're at least not in the first pass of our understanding of this. Similarly, we took a peek at this for cannabis use, and again, you'll see here that there was some movement in the main proposed mediators, or motivation to change had some action, as well as self-efficacy. 2, we had treatment effects where MI led to reductions in pure norms in the right direction, but then you will see that that did not actually correspond with reductions in marijuana use. Excitingly, we did have a main effect for MI in terms of better outcomes, but you'll see here too that motivation to change, more motivation change led to more marijuana use. And then more, here we had self-efficacy in the right direction. So what you'll see again is that we are really wrestling with what these mechanisms are and how they might affect adolescent substance use. And we are not there yet. Hopefully we'll be there soon with discussions that we're having with this data, but it is muddy at best in terms of trying to understand it. So one of the things I've been thinking about a lot as an active clinician is what's happening in the room. I think about my interactions with kids when I'm meeting with them and trying to think about what we can do as therapists that are impacting adolescent outcomes. So in my early studies, I thought surely empathy and therapeutic alliance would be the main thing that causes it. Empathy and MI's greatest strength seems to be that it very quickly causes a big growth in therapeutic alliance. And so you'll see here, again, this is early work showing that MI outperformed the control in terms of binge drinkers with emerging adults, but there was no significant relationship for empathy and therapeutic alliance. So 2, I thought maybe the money is in the ethnicity. So here we looked at Hispanic versus white kids. This is in the big R01 that I just showed you. In fact, Hispanic kids did have greater alcohol-related problems at 3 months, and this was actually mediated by reductions in therapists' behavior. So therapists showed less MI spirit, less supportive autonomy, and less complex reflections with Hispanic kids. But interestingly, that only related to poor outcomes for alcohol-related problems. So there was no difference in outcomes for cannabis-related problems, cannabis use days, or alcohol use days. So I keep coming back to, are there better, more sensitive metrics that might help us understand sensitivity, specificity, and the therapeutic process, as well as addiction treatment outcomes? So one factor that I had really been thinking about is maybe neurological factors. In some early work I did with Kent Hutchison, I looked at the role of depression and anxiety, finding that, yes, sure enough, greater depressive symptoms and greater anxiety symptoms led to greater cue response. And this is an gustatory cue. Similarly, more recent work was looking at a go-no-go task with adolescents, and we found an inverse relationship between response inhibition and substance use days. I've also been thinking about the potential role of basic biological factors, including genetics, as mechanisms of treatment response. And here again, this is earlier work showing that DRD for long individuals had poor outcomes in MI, or which was motivational enhancement therapy, versus the alcohol education control, as compared to kids who had the DRD4S genotype. So, this has really caused me to think about, trying to link biology in the brain in terms of adolescent decision making. And really, this brought me to Francesca because I keep coming back to the brain as a vehicle of change. As a parent, I often say to my kids, you need to use your head, you need to think about things. And we also, we all presume that when we're having conversations or working with therapists that we're thinking about things, that this change is occurring in our brains. But somehow we often overlook the brains as a place to think about in terms of behavior change or in terms of therapeutic process. So, in the early stages of my collaboration with Francesca, which I should actually give credit to Kent because this all occurred in the context of his lab, we took his some work that he had been doing with taste exposure, and so I wanted to use something that looked like it really worked. And so he had really good the work that he and Francesca had been doing had really good findings in terms of alcohol cue response, and I thought, it was so clear the difference between those with alcohol dependence and those without, or controls. I thought, I wonder what would happen if we took a very strong, empirically supported mechanism of change in the MI literature, which is change talk and counter change talk, and interspersed that with this gustatory cue paradigm. And for those of you who are not familiar with change talk and counter change talk, which is now called sustained talk, the world, there's been a really strong literature showing that these two different types of client language predict outcomes as far out as 2 years after treatment. And so, but I have never ever been clear, either in the room or out of it, or as a scientist, on what change talk and counter change talk really are. What's happening in the brain as those things are happening? Is change talk a proxy for behavior change? Is it a kind of a signal for what's happening in the brain? I don't know. And so is it a shift that's happening in the brain? So I just wanted to get a better sense of what was happening there. And so what I did was meet with 10 alcohol-dependent adults. I did the MI with them, and then I took their statements, change talk and counter change talk statements, from their session with me, and we played it back to them in the scanner. So they heard their own statements in the scanner, as well as saw them. For those of you who've not been in fMRI, it's noisy. It's like being in a very strange, noisy, weird tube. But so in terms of ecological validity, we talked about this a little bit yesterday. I wanted to make sure that they could see but also hear themselves. We do also test to make sure they can hear the voices before we start the session. So we played back their own statements back to them and then gave them the cue exposure, which is a taste of their favorite beverage alternated with taste of an appetitive cue like lychee juice, which tastes sweet but which they've often not had before. And here's what we found, a very striking contrast with when people heard their change talk, these are statements in favor of changing their behavior, there was no activation, and when they heard statements in favor of staying the same, continuing to drink, that they looked much like people in standard cue exposure paradigms. And this contrast was robust. So, when this is change talk greater than counter change talk or sustained talk. And here you can see, when we contrasted the two, this is reasons in favor of changing over reasons in favor of staying the same. Reasons in favor of staying the same over changing, that there was quite a bit of activation in all the key mesocorticolimbic centers that matter in terms of alcohol dependence. So we were really excited about that. And then this actually, I'm going to say how this works in terms of concrete processes. So then I went to Francesca and said, what's happening? What is that? And so, I really wanted to, as a clinician, draw I sort of had this model in my mind of the relationship between what happens in the room, and then I also often think about what happens. So we as therapists think, like, we must be so powerful, right? But people are in the real world with friends who give them drugs or partners who give them drugs. And so I am also cognizant that what I say as a therapist, it might not have that much staying power once somebody's out in the real world. And so it's always important to me, this connection between what somebody says in the room and then what happens out in their real lives. So, I kind of sat down and thought about all the different things that I thought might be happening in the brain, both when people were talking to their therapists and when they were out in the world, and then went to Francesca and said, where are these things happening? So this was the beginning of a dialogue about all the things that could be happening in these clinical interactions and outside. And I think it's really important to have an expert who knows the other side of that better than you do, or better than I did. So the first comparison that I just showed you, the action really seemed to be happening right there in this model. We then extended it to adolescent cannabis use. So we took Francesca's paradigm looking at cue exposure, and again, this time I used 10 different therapists. I trained up a bunch of graduate therapists so that we had a little more ecological validity, and played back the same sort of thing, change talk or sustained talk, from their session right before they went in. I want to present to you a little more detail about this sample because, I was just at CPDD where the average age of onset of cannabis use was 16. Here you'll see in our justice-involved sample, it's 12. So the kids that I'm working with, these justice-involved kids, are much higher risk. You'll see 97% of them are using during weekdays, which is a good predictor of interference in school. They have 18 cannabis use days, which is almost parallel to Lara Ray's methamphetamine users from yesterday. These are kids they have very high rates of cannabis problems, cannabis dependence, and pretty moderate rates of other substance use, which is very standard for kids who tend to be polysubstance using. I also, this is for the clinicians in the room, want to show at the 1-month follow-up, after their session, they showed significant reductions in cannabis use days, cannabis problems, and cannabis dependence. And this is a clinically significant metric of reduction, as it's a halving of symptoms across these metrics. So here I show you, this is again, just for the sake of comparison, the adult data, and here I show you the adolescent data. So you'll see, in contrast to the adults, where there was no activation in change talk, here all of the activation was in change talk, with no activation in counter change talk. So kids were thinking more, or there was more activation when kids were thinking about changing, and this is thinking about changing over thinking about staying the same. And you'll also notice that these areas are more areas that are important in introspection and contemplation rather than reward. We also want to know how well do these data correspond with behavior change. And so here, this is the relationship between cannabis problems and activation at baseline and 1-month outcomes in cannabis problems. And you'll see here, greater activation meant during thinking about change led to better outcomes. And similarly here, there was more activation led to better outcomes at the 1-month post-treatment. And I'll also highlight the precuneus. So the precuneus keeps coming up as an area that we didn't expect, but particularly in adolescence, it seems to be a key player in terms of behavior change. So this gave us another perspective on our model, both in terms of what's happening in the room in terms of change talk, as well as what's happening in terms of after they leave the therapist's office. We also link this to genetic factors to see, if we kind of go back in terms of going down basic biology back to genetic factors, will there be a relationship with therapeutic process? And indeed, for the serotonin receptor polymorphism, HTR2A, kids who had the CC versus the T allele groups had more activation in change talk versus counter change talk or sustained talk. So there does seem to be an even earlier, more fundamental basic biological factor that's predicting or that's correlated with treatment response. So another factor, I told my old mentor Bill Miller about these data, and he said, oh, well, what would happen, I wonder if this is just the act of saying these things to yourself. Is it just saying those statements over and over again that's causing this activation, or is it really due to the therapist session? So I thought, Let's find out. So I took a group of college binge drinkers, and I took half of them, and we gave them the same paradigm that we did before, 2 sessions of MI, where we extracted their statements and presented them back. That's self-generated. And then we took the top 5 statements from that group and presented them to the second group, experimenter selected, where we just had them read the statements and then put them in the scanner. So they were controlled. Both groups got their own voices and their own statements. One had a therapy session, one did not. One had the statements generated from a therapy session, one did not. And here's what we found. Sure enough, the self-generated group, when you generate your statements in the context of a therapeutic session, you have more activation in these areas than when you just read them off of a sheet of paper. And in these critical, important areas, including the IFG and insula, as well as in areas like the supramarginal gyrus. The same thing, we also found activation for sustained talk, though, that it also seems equally important to have your statements come in a true therapeutic session than to have them just saying them. It's not, you can't just talk yourself into change, apparently, because there's less, that'd be great, I guess, but what we found is there's more power in generating these things in a meeting with a therapist, also in these important frontal, temporal, and parietal areas. So that gave us some sense of just kind of a preliminary view of that there might be something happening in this within session process, particularly with the therapist. So we then have been thinking about, is there something happening in this therapeutic relationship in terms of behavior change that's mediating those outcomes? And this is actually the foundation for a new R01 that I just received. Thank you, NIAAA, and we'll be beginning shortly. And this is just, this is actually we did some preliminary looks at that potential mediational relationship, a correlation before doing the mediational relationship, looking at complex reflections. Again, as a therapist trained in motivational interviewing, you're told over and over again of the importance of doing these very hard, sophisticated, complex reflections, which is, reflecting back some emotion or trying to further the conversation in order to elicit change talk versus closed questions. So those are sort of a no-no in MI, where you do things like say, how many drinks have you had? Has anybody told you about your problems with drinking? And so we looked at so we played back therapist statements to kids in the scanner, and we found that for complex reflections versus closed questions, there was difference in activation, particularly in the ACC, and that it was significant. So that has given us some sense of these relationships in that process. So one thing that we have been really thinking about, and I think it's important for neuroimagers and clinicians to think about, is this issue of replicability, right? So we have also, it's very exciting that we've been doing this, but is it the case that if I do this with a number of different studies, that I'm going to find different things? How much overlap is there? And one neuroimaging method that allows you to examine the degree of overlap is conjunction analysis. So it's not quite meta-analysis, but it's kind of a way of just looking at how strong the results are across studies. I was particularly interested in the adolescent data, I think just because it seemed to be so different from adults and just because it's an area of my interest. And so we really wanted to see the degree of neural overlap in these studies and, to see if this could inform common pathways underlying treatment response with substance-using youth. I was also interested in the comparison of marijuana-using kids, but also these adolescent binge drinkers. Was there some difference in the response because of the substance of abuse? So we added these 2 data sets together, and here's what we found. So this is with the crosshairs centered on the IFG. This is the combined 2 data sets for alcohol and marijuana, and you'll see quite a bit of activation, and also quite a bit of activation in the precuneus for change talk, and this is for sustained talk too. You'll see almost parallels levels of activation, which is compelling in the IFG as well as in the precuneus. So we did then, I thought maybe there was going to be some relationship by age or gender. We did not find differences by age and gender, nor did we find differences when we compared change talk with counter change talk or sustained talk, and sustained talk in contrast with change talk. So this really gives us some sense of what might be happening. You know, when I talk to people about these data, they always say to me, yeah, that's great, but you know, what does this mean for on-the-ground practitioners? And I think there's, you know, two levels of response to that. On the one hand, these data have actually made a difference in my clinical practice. So I do different things because of this work. I allow more time for the kids that I work with. I've changed the way that I write manuals in order to give kids more time to think about their substance use. And by think about it, I will say, most kids in our studies say that they've never even thought about how their substance use could be beneficial or detrimental to them. It's really their first time reflecting on substance use. They just have been using without ever stopping to think about it. And so I think that might be where the magic happens in MI with kids. So we have been adapting the intervention in order to really bolster that part. At the same time, we are still very much in the preliminary stages. You know, I really do, especially for you MOBCers, you know, the work in how change talk and counter change talk, how that impacts long-term outcomes does inform the way that we write and create manuals. And so we are at the very ground level, but we are taking the same approaches. So what's happening in the brain, you know, somebody said yesterday, does it matter if there's no change in the amygdala? No, it doesn't matter. But if we know what's happening, if the amygdala, for example, is where the money is, then we know how to tailor interventions to enhance those things. Does that make sense? So there's really a back and forth, I think, between the brain and behavior that we ought not ignore and really use to inform how we work with kids to make things work better. The other thing that I'll say here is, you know, we spent a lot of time yesterday thinking about the models in adults, and the models for kids are different. The adolescent brain is not the adult brain, even though I wish it were, right? It would make things very simple. And the toddler brain is not the same either, as I'm finding as a parent. But so I think it's very important for us to allow to be informed. We should be informed about adult models, but we should also not be bound to them. So I think it's very important for us to think about how these treatment mechanisms might be different in the adolescent brain, so that we can understand both the process of addiction, as Lara mentioned yesterday, but also the process of development. And that's complicated because we also have to think about the process of change in those contexts while these other things are happening. So I just put that out there as something to think about. I also will put a plug for Francesca and I have also been thinking about TMS and trying to think about if we can modulate the adolescent brain to enhance treatment response in the context of MI with cannabis users. And finally, I end with some work that Katie, Francesca, and I have been doing. We have a collected volume on neuroimaging and psychosocial addiction treatment, much of which highlights work that has been presented throughout the past 2 days, and it is now available, should anyone want to read more. Thank you so much. Oh, so now we have time for questions. Questions?